Yeah, as John said, I'm the new member of this group, I guess. So we have met in
We
France.
have a few new members.
Yes, all right. What I'm going to talk about is obviously we haven't worked with the Sin 3 or versus the earlier version either, but so I will plan out now what our plans are. And hence I'm presenting one of the current activities that we have in the lab. And I think it fits nicely with the things that we want to do. It's a bit of a mouthful title, but I will walk you through. It's briefly, as John said, I'm from Norwegian University of Science and Tech. So we're in this town called Trondheim. We are pretty high up in the map that John has shown. And my lab is part of the Department of Biotech. It focuses on biophysics, computational, and synthetic biology. So one of the focus areas in my lab is studying transcription and translation regulation is actually across a wide range of microorganisms. Currently, we have more than 13 that we work in the lab. I may show a gene expression cassette, if you will, promoter 5' UTRs or coding sequence in this fashion. But we rather like to look at it more like this. So even though it's the DNA, when you look at the functional level, there are many functional units that are present both at the DNA or maybe even at the mRNA level, and even if so, in the protein level. So we have two types of approaches. One of them is the more on the understanding level, and on the controlling level, we also have some startup that we use AI type of approaches for designing of sequences. But today I'm going to talk about our efforts in terms of understanding and especially focusing on the sigma factors. And also in terms of motivation, I'm sorry, this is not from neither of the minimal genomes. The resolution was too low, so I had to borrow another image. But it conveys the message is that when we do genome annotations, all we do is annotating coding sequences. So we don't know much about what regulation applies, that all the genes that are present at the genome level. And one of our motivation is actually focusing on and mapping the sigma factors. So what we landed on is working with these three specific microorganisms. So Bacillus subtilis, E. coli, and Pseudomonas putida. Bacillus is a gram-positive, E. coli and Pseudomonas are gram-negative. They are maybe not really spanning really high GC content, but it's 43 to 61%. But one unique aspect they have is that actually these guys have different sigma factors. So E. coli has in total 7 sigma factors. Bacillus has 18, whereas Pseudomonas has 24 different sigma factors. And in our current efforts, and I'm going to tell what we are doing, we have now mapped the sigma factor binding sites, 17 out of 21 different sigma factors in our project. The protocol is as following. First, we generate quite a large number of plasmid DNA libraries. And this is currently in the number of about 4.5 million unique DNA templates. And once we isolate this DNA, we sequence the whole content. Hence, we know every one of those 4.5 million DNA templates. And what we do is that of the 17 sigma factors, we take one sigma factor at a time and take the corresponding core RNA polymerase from the organism. So this image only depicts the path for sigma 54, but you can change it to either of the two organism and the corresponding sigma factor. But the procedure is the same. We take this large number of DNA templates we have, and then we run in vitro transcription in the presence of one sigma factor at a time and using the corresponding core RNA polymerase. And from there on, we do perform DNA and RNA isolation. Obviously, the DNA isolation and the sequencing determines us the composition of the templates that serve in the IVT reaction. And once we isolate the RNA, it's obviously these are the functionalized RNAs. And there, I want to add one element there. So when we make this when we make these large or extensive libraries of 4.5 million that I have mentioned, we are using an RNA aptamer, and that is so that once it is transcribed, and if there is a fluorophore present in the reaction, it gives a color. And so that means when we perform transcription or in vitro transcription in the presence of one sigma factor at a time, we collect the RNA based on the signal, but we also collect just for a pure RNA isolation level. So we can determine by using the signal of the intensity level, and I will come back how we use that one. But in this in vitro transcription specifically, we only collect all the RNA that are generated. What that means is that if there is an RNA generated, that means there was a corresponding template present in the large pool. And we do perform RNA isolation and sequencing, and then we end up is that DNA sequence versus mRNA, and then we used 5' UTR as a bait to map which promoter actually served in the immunotranscription reaction to generate this particular mRNA. Here I use the term D. broccoli because this is a bit of an old image. I apologize, it should have been mango because we use different aptamers throughout the process. But what this approach allows us that, so in this particular case, we have 1.45 million DNA templates that served in the reaction. And of these, we could generate about 64,000 promoter sequences we could identify serving as template in immunotranscription reactions. And when we do RNA isolation and sequencing, we perform also a 5' enriched cDNA sequencing effort, and that allows us to determine the transcription start site across these templates. So with that, we are generating a really high resolution data specific for the sigma factors that I have mentioned. And now I'm going on beyond the sigma 54 because this was the first sigma factor we worked with. Currently, the numbers are we have 4.5 million unique DNA templates. And of these, we have about 1.5 million functional RNA sequences. And this is corresponding to all sigma factors that we work with. How do we go from here? And obviously, we are doing both in vivo and in vitro validations. We also take the genome sequences, chop them into small pieces, run them through the same procedure instead of using our libraries, and there we explicitly nail it. And with all the information that we are generating, we are currently updating the genome annotations to add the regulation into the annotation picture. As you may recall, when I showed you the previous or one of the earlier slides, currently we only annotate coding sequences, but our intention is actually annotate the regulatory sequences as well. And one of the things that we are excited about is that because currently there isn't really a go-to tool for promoter prediction, especially for the bacterial world, by using this really cool data that we have now, and it's pretty extensive and large scale, we are training deep learning models, and I can tell that actually the models are performing really well. Currently, we are working on the model development, but once it is ready, we will also develop the web-based tool so that everybody can go and paste their sequences and hopefully get some decent predictions out. Given the time limitation, I decided to focus on the last aspect of continuation of these efforts, the microfluidic-based phenotype genotype characterization. Obviously, when we run the immunotranscription reactions in bulk, we don't get the genotype-phenotype correlation because there are many templates that serve as template, and then at the end we get total RNA. Even though we could read the aptamer intensity, it's not one template, one phenotype. In order to get one construct to one phenotype, you're performing in vitro transcription in droplets. So what we do is that we take those 1.5 million, or depending on different libraries we have, so we could take the entire library as well, and we generate droplets that have one template per droplet, meaning we have quite a fraction of empty droplets, but we are happy with that. And then we're running DNA amplification based on isothermal amplification to increase the copy. And thereon we perform in vitro transcription reactions. And the pictures looks like this. And again, what we do is that we identify those droplets with different intensities. We also bin them. We can't unfortunately go down the one template, one drop, one phenotype readout, but we nevertheless bin them, and that gives us a much higher resolution. So what we're trying to do there is that to achieve the genotype-phenotype relationship, giving not just the, a sequence serves as a promoter or not, but also depending on the sequence composition, how much activity it leads to. If there were some questions, I made some slides that I could explain through the procedures. So this is practically the acknowledgement slide, and then maybe while leaving it, that I can maybe go back to tell what the relevance is. I think in John's slide there was number first. So we want to now continue in our efforts in terms of understanding how transcription regulation happens, especially at the minimal genome level. And maybe the key reference here is that, John, please correct me if I'm wrong, there is no sigma factor left in the minimal genome. Currently, it's only one, but nevertheless, the organism still manages to maintain its transcriptional activity across the growth stages. And if you look at the literature now, everybody thinks that different sigma factors allow that one. So in a way, when you reduce a sigma resolution to a one sigma factor only, so that means that a lot of auxiliary proteins must have been involved in this process. And maybe you can even think of some time signatures, not just that a certain promoter determines the strength, but maybe you even have signatures such as when something should be turned on and on what level, and maybe all the proteins that are involved. So we're trying to understand the basics of the transcriptional regulation using the minimal genome. So that would conclude my presentation, and I'm happy to take questions.
Sorry, questions for Rahmi? I'll start. So, Rahmi, we sent you data for the transcription for the wild type, the JCVI SYN1, which is ostensibly the wild type Mycoplasma mycoides, which again only has the single sigma factor. So have mycoplasmas been able to throw away? I mean, I would speculate that mycoplasmas may have started out like a Bacillus subtilis with a lot of sigma factors, but as part of their genome minimization through massive gene loss, they may have been able to manage transcriptional control with with many fewer transcriptional regulatory proteins. Any thoughts about that?
I mean, the short answer is no, I don't know as of now. And that's what we want to study, because what we do know is that there is a lot of auxiliary proteins involved in transcription regulations, which we are not considering, including translation as well. I mean, S1 protein is one of them. You can have leaderless sequences, but still manages to identify the genes, and you get translation running. So I do not know, short answer, but long answer, we want to study, search for different signatures. How could they maintain these transcriptional profile over growth stages and still have a stable growth? Not like every individual cell behaves differently. That's not the case, right? So there is some sort of phenotype and is reproducible in a way. But with the current transcriptional model and transcriptional regulation understanding we have, this should have not worked. So hence there's a lot of missing elements, and that's our intention to discover that.
I know we sent you data, but I don't think we sent you cells, did we?
No, so I will come back to that one. I will bother you separately. I mean, in this presentation, of course, I didn't talk about the main reason that we have gotten the data, but maybe in case somebody is curious. So we also have a company or spin-off where we do predictive DNA design. So that means giving the context, if you have a promoter 5' mutator coding sequence, we have an algorithm that in one way or another predicts transcription and translation rates. So we want to just add that one as a higher result to get a bit of a higher resolution in terms of what type of regulation do we foresee giving the transcript profile, because we want to get hold on to the cells and do some transcriptomics ourselves. As I said, especially due to the 5' enrich to determine the transcription sites. We have some ideas, because what we have seen is that transcription start site organisms have, for example, we have seen is not a single one. So maybe there's one other level of that shifting that one allows some level of control. So the data, we couldn't really resolve it as of now, but I will come back to it. And of course, we would like to request the strain as well, but I will bother you another email on that. Maybe as a to conclude, I'm a new member, so if you're curious about any of the things that we do, please reach out. Otherwise, I think I will keep my collaboration limited to John.